last time we have seen that this electron transport chain, it transports horizontally electron while horizontally across the inner mitochondrial membrane with the transport of electrons. The protons, they are pumped out of matrix in a inner membrane space. We have already seen what is the total energy, useful energy liberated. Have you calculated it? How much it is? 46 into 1.14 is 152. So that's how the energy is liberated, and what you get is pumping of the protons across inner mitochondrial membrane. All complexes, complex 1, 3 and 4, they participate in the pumping, or they pump protons from inside the mitochondria to the inner membrane, from matrix to the space, intermembrane space. So we have already seen that there is a outer membrane, this is inner membrane, we have magnified for our convenience and understanding, and this is matrix. Okay, and finally the electrons that are passed from the or reducing current that are passed from the substrates in the form of NADH. Okay, converted to NADH. NADH is converted into NAD, and reducing currents in the form of protons, electrons, hydrogen ions, they are transported across electron transport chain. Finally, it meets with the oxygen. Oxygen is reduced to the water. Now, while doing so, there is pumping of the proton, and the energy is liberated, and that energy is, we know, is directly proportional to the, or we know the formula, that is delta G is equal to NF delta E. It comes about to 52. So let us see what next happens in the electron transport chain. We have seen the mechanism by which electrons are transported and protons transported across the inner mitochondrial membrane. So this is the membrane. Now we know that this is impermeable. So whatever protons that are transported out of the matrix, they cannot easily enter inside. Okay, because this membrane is impermeable. And due to the pumping of these protons, there is gradual positivity more as compared to the matrix. So you can say that it is positive out, negative in. So this creates a gradient, potential gradient. So less positive is in the matrix, more across the membrane in the inner membrane space. So there is formation of a gradient, potential gradient, plus these are all protons. That also creates another gradient, that is pH gradient. Okay. So there is difference of pH and difference of potential. So this is called as electrochemical potential difference. So there is electrochemical potential difference is there, and this difference is called as electromotive force. Okay. So this is a force that is utilized for the next step. So there is formation of electrochemical, means more positive, that is electro, and more acidic outside, that is chemical. So both way there is a difference of positivity, negativity means more positivity across the membrane in the inner space, as well as there is more protons, so acidity is more there, or acid pH is in the inner membrane space than in the matrix. So that creates a potential difference and electrochemical potential difference. Electrochemical potential difference, it occurs across the membrane. Now we have seen that there are 4 complexes of electron transport chain, and there is another complex that is described, that is called as 5th complex, complex 5, and it is named as ATP synthase. Okay. As ATP synthase, if you remember the mitochondria, it is seen as on the inner mitochondrial membrane, it is seen like a lollipop. An electron microscope. I hope all of you know what lollipop is. So it looks like lollipop. So there are 2 parts, one that is embedded in the inner mitochondrial membrane and another one which is floating in the mitochondrial matrix. So likewise we can see this is what it looks like. This is simply, we'll draw this in detail afterwards. So this particular part is called as F0, actually it is O, to denote that it is sensitive to oligomycin, and this part is called as F1. Okay. So there are 2 parts, F0, F1. F0, F1 particles, they made this particular ATP synthase. This is called as F0 ATPase, and this is called as F1 ATPase. F1 ATPase is more common name because its activity is to hydrolyze ATP. If this part is separated, it will hydrolyze the ATP. That's why the name F1 ATPase is given. And this total complex is called as ATP synthase. Now this ATP synthase, it has got some structural subunits in it. It is known to have alpha 3, beta 3 units, alpha 3, beta 3, delta, epsilon, okay, A, B2, Cn, 9 to 12. So this is the configuration of this particular ATP synthase, that it has got 3 alpha units, 3 beta units, 1 delta, 1 epsilon, 1 A, 2 B units, and 9 to 12 C units. So this is how this particular segment is, or this particular F1 ATPase. Now for our convenience to know that this is the matrix side, I am drawing this thing upside down. Okay. For your convenience, you can draw it like upright. Okay. So this F0 segment, it constitutes the C part. So it has got number of C units in it. These are C units. Okay. With this C unit, there is a delta unit which is attached to this C unit, and this delta is slightly, you can say, like a bent axle. Okay. It is just like a bent axle, because this is finally is the smallest miniature motor known to us. Okay, this works as a motor. We will see it later on. So this is the gamma unit, which is attached to the C unit with, sometimes they say that it has got some delta unit or epsilon unit, which forms or which attaches this gamma unit to the C units of ATP synthase, plus it has got 3 alpha and 3 beta units. So it has got 3 alpha and 3 beta units. Bear with me my drawing. 1, 2, 3, 4, 5, and all like 6. So there are 6 units around the gamma unit. You just forget the gamma unit from inside. That makes more complex. Just remember that there is gamma unit inside it. So it is not attached. What attached over here is gamma unit. Small gamma unit can be seen over here like this. So there are 6. This is alpha. This is beta. Alpha, beta, alpha, and beta. These are the units. So there is a bent axle inside, around which the 6 alpha beta units, 3 alpha and 3 beta units, it forms a structure. Now this is held to the cell membrane with 2 units. These are so this is A unit and these are 2 B units. Okay, that is linked to the C units. These are the B units. Now this B unit holds this particular 3 beta and 3 alpha units with delta. So if you draw it like this, so you will just see like this, that this is the alpha beta units, this is C unit, these are the A and B units, and this is bent axle inside, that is gamma unit. This is how it is seen over here. ठीक है? Have you got it? So the particular ATP synthase is a unit which has got 3 alpha, 3 beta, delta, epsilon, A, B, C, and numbers are there. What remains over here is gamma. It is a bent excel. So actually you can write how many units it has got? 13. 3 alpha, 3 beta, gamma, delta, epsilon, A, B2 and C 9 to 12. So numerically it has, or subunit numbers of ATP synthase are 30. Alpha, beta, gamma, delta, epsilon, A, B, C. These are the units of, so total number is 13. Out of these 13 units, remember that 4 units, they are encoded by mitochondrial DNA. Out of these 13 units, 4 they are encoded by mitochondrial DNA. Now what happens next? This is, we know the structure of ATP synthesis. What happens exactly in the, how ATPs they are synthesized? This is just, we know that everything is happening, electron is flowing, finally water is formed from oxygen. But where are the ATPs? Show me the ATP. How ATPs they are synthesized? Because nothing over here is seen to be capturing energy in the form of ATP. We already know that there are two ways by which ATPs they are synthesized. One is substrate level phosphorylation. We know the examples, 2 examples in the glycolysis and one in the tricarboxylic acid cycle. So directly where synthesis of ATP takes place at that in substrate level. But here, as electrons are transported from donor to acceptor in a stepwise manner, finally to form water. But how this is linked? And this was the problem to which scientists they just struggled for so many years. How this particular thing this occurs. And then in 1960, one of the answer to this question is given by a scientist known as Peter Mitchell or Michel. In 1960, he put forwarded the exact theory of synthesis of ATP. Before that, what we just know about, or what was speculated, that some intermediate is formed during the process, and that intermediate then is helpful for the synthesis of ATP. But that intermediate was never been found. So this convincing theory was put forwarded by Peter Michel in 1960, which describes the synthesis of ATP, where two processes they are coupled. The process of electron transport is oxidation, and process of ATP synthesis is phosphorylation. So these two things they have to be coupled, and there was no mechanism before known how this particular thing occurs, and Peter Michel put forward in 1960 a theory that is called as chemiosmotic theory. Chemiosmotic theory of ATP synthesis. Chemiosmotic theory of ATP synthesis. Now what this theory says, or what it proposes, we already know that there occurs a difference. Which difference? The difference due to pH and positivity, that is electrochemical potential difference, and this is electromotive force. Now this force is used, this energy that is liberated is used to pump or to send these protons which are in the inner membrane space back into the matrix, because inner mitochondrial membrane is impermeable. So these protons, they sent through this C units, okay, inside the mitochondria. This particular electrochemical potential difference is used to pump or to again send back the protons inside the mitochondrial matrix. Now if you know the mechanism of windmill or water mill that is present at Aurangabad, Panchakki. So the force of water is used to drive or to get a flow. Okay. So that particular force which is putting on the turbines, that rotates the turbines, and due to the rotation everything occurs. Next, so same thing occurs. So there is entry of protons inside, and these C units, they can just move like raketic movements of C units. It occurs within the cell membrane. So whenever proton comes inside, it is rotated. Next, so likewise this particular thing, it rotates. Okay. Now if this rotates, what happens? There is a gamma unit attached to it through epsilon or delta. That also rotates. Okay. Are you getting me? That electrochemical potential gradient across the membrane is used to send back the protons through C units of ATP synthase, and it enters inside with that motive force, electromotive force. The small C units, they move or they just rotate rather than movement. They rotate, okay, in the cell membrane. As they rotate, the thing that is attached to it, it also rotates. Okay. Now this is not symmetrical. Gamma unit is not symmetrical. It is bent like a bent axle. Okay. Suppose these are 5 fingers. You can have anybody sees. So this you can suppose to be a ATP synthase. Okay, 3 beta units and 3 alpha units, and there is a bent axle over here, or just like a fist, and then there is a bent axle inside, and these are the C units, and it is there is occurring movement. So what happens? This rotates. You can feel yourself. Make your fist and rotate your bent axle, your finger, index finger inside. What happens? What do you feel inside? Yes? What you feel inside? If it is straight, then you don't feel anything. If it is bent, then you feel pressure inside. Anna? You feel pressure inside. And this is the pressure. Same pressure, it is felt by alpha beta units. Or you can say that due to this, what happens? There occurs conformational change in beta units. There occurs conformational change in the beta units. And these are the beta units which are important for the ATP synthesis. So there are 3 conformations of beta unit. This was put forward by Paul Boyer, okay, for which he got Nobel Prize afterwards. So this movement or rotation of gamma unit inside the 3 alpha 3 beta units leads to the conformation changes in the beta units, there are 3 beta units. They have got 3 conformations. One is tight conformation, another is open, and one is loose. Okay. So what are the functions of these 3? You know how enzyme works. This is ATP synthase. This is enzyme. What is the thing which helps enzyme to work is the formation of active site and catalysis. Why enzymes they are specific? Because of their active site and because of their structure. Same thing, ATP synthase is an enzyme. It has got specificity as well as the catalytic activity, and it depends, this catalytic activity depends on its conformation, the structural organization of protein or enzyme. Enzyme has got a structure, 3-dimensional structure, and that structure is important for the catalysis. And this particular thing occurs inside from inside, so there occurs conformation change. So from loose to tight to open. So from loose, where ADP and inorganic phosphorus are there, it get converted into tight, where catalysis occurs and ATP synthesis it occurs, and when it opens, ATP is liberated. Are you getting me? So here ADP and inorganic phosphorus, it enters, and ATP it leaves the beta unit. And all this occurs due to movement of the bent axle. Bent axle moves due to the movement of C unit. C unit moves due to the entry of protons or hydrogen ions through it. Entry of hydrogen ion occurs due to the electrochemical potential gradient, and electrochemical potential gradient occurs due to the pumping of protons in the inner membrane space, and this pumping occurs due to the transport of electron across electron transport chain. So this is how 2 things they are coupled: oxidation, phosphorylation. So these 3 tight, loose and open sites, they are alternate in 3 beta units. So during a single turn, how many ATPs will be formed? 3 ATPs will be synthesized. Okay, 3 ATPs will be synthesized during one turn of bent axle. Okay, so these conformation changes, in short, again through electron transport chain, the electrons, they are pumped across the inner mitochondrial membrane, which is impermeable to the electrons. Due to the pumping of this is impermeable to the protons, and due to the pumping of proton through the complex 1, 3 and 4, there is formation of electrochemical potential difference across inner mitochondrial membrane. This is electromotive force, which is used to pump back the protons through the ATP synthase C units. C units rotates, with it gamma unit rotates, which causes conformation change in the beta units of ATP synthase, which leads to ATP synthesis. This is oxidative phosphorylation. So oxidation is coupled with the phosphorylation. Okay? This is how oxidation coupled with phosphorylation. Oxidation do not go itself without phosphorylation, and phosphorylation cannot occur without oxidation. So oxidative phosphorylation is the process, as comprehensively said, we can say comprehensive, like electrons, they are transported from donor to acceptor in electron transport chain. in a stepwise, controlled manner at body temperature, at body pH, as per body need, to form a electrochemical potential difference across inner mitochondrial membrane, which is used to pump back the protons across C units of ATP synthase, and due to which there is formation or synthesis of ATP. This process is efficient and called as oxidative phosphorylation. ठीक है? So oxidative phosphorylation is stepwise. It is controlled. It is at body temperature. It is at body pH. It is as per body need. And not only that, it is efficient. Why we call it efficient? Because of PO ratio. How many ATPs they are synthesized per molecule of oxygen? It is around 2.5 if substrate enters at the level of complex 1. So from NAD you can say that if substrate enters at the level of NADH, you get 2.5. That is, if substrate enters at complex 1, you get 2.5 ATPs, and if it enters at FADH2 level or complex 2 level, you will get 1.5 ATP. Before that, it was said to be a 3 ATPs and 2 ATPs, but nowadays we say that it is around 2.5 ATPs and 1.5 ATPs. They are formed when electrons they are passed from complex 1 and complex 2. So how many ATPs you get when there is a drop of around 50 kilocalories? What is expected and how much is formed? We have calculated around 52 kilocalories liberated, okay, delta G. So how many ATPs you expect? Around 7, but you get 2.5. So how much is the percentage? 2.52. So around 30% is the efficiency of electron transport chain. Remaining energy is dissipated as heat and may be required for the maintenance of body temperature. So this is all about the oxidative phosphorylation process. Oxidative phosphorylation process. And mind well that these are very much tightly coupled. They are very much tightly coupled to each other. They cannot be separated unless until there are certain physiological agents like something which causes short circuit or which causes entry of proton inside the matrix. So it will not lead to formation of ATP. So protons, whatever protons pumped outside, they will come inside, and energy what is liberated is used for the heat. And this is seen in brown fat tissue, brown fatty tissue, where thermogenin is present. Thermogenin is seen in brown fatty tissue or brown fat. So there are two types of adipose tissue, brown and white. So brown adipose tissue, it is seen in infants. Okay. Later on, as you become adult, the brown fat vanishes. Brown fatty acids are replaced by the white adipose tissue. So in brown fats or brown fatty adipose tissue, there is a protein called as thermogenin. Now this thermogenin allows proton to come inside the matrix. Okay, so you can just say that there is a formation of hole in the mitochondrial membrane. So due to that, what happens? There is no electrochemical potential gradient, or there is no difference. That's why ATP may not be synthesized. What you get is just heat, some of the energy that is liberated in heat. And this is very much important, because you know, infants or children, what happens in the cold nights? Everybody is wrapped in blankets, and you try to wrap the infants in the blanket, they just kicked off it off, है ना? So mother and father they try to wrap them in the blankets, but they just throw it away. But nature has given a protective covering with the help of brown adipose tissue, which generates heat and which keeps the temperature maintained, because whatever energy is liberated, that is used as heat, and body temperature is maintained or is raised. So this is thermogenic, and this uncouples oxidation from phosphorylation. This is called as uncoupler of oxidative phosphorylation. This is the uncoupler. Then there are other uncouplers like thyroid hormone also uncouples, and there are certain chemicals, dinitrophenol, cresol, which uncouples the oxidation and phosphorylation. So these are called as uncouplers. Uncouplers means where oxidation goes on without phosphorylation. First thing first, you have to keep in mind that oxidative phosphorylation, where two things, they are tightly coupled to each other. One thing cannot happen without other thing. So oxidation is there and phosphorylation is there. So only when some chemicals, or only where the physiological uncouplers are there, then and then only these two things, they are going independently, or you can say that oxidation is going on without phosphorylation. There is no phosphorylation. So this is uncoupling. Apart from this uncoupling, there are some inhibitors which inhibits oxidative phosphorylation, the process itself. The electron transport chain complexes can be inhibited by the various you can say reagents, chemicals or poisons, and there are dreadful effects of this inhibition of oxidative phosphorylation. What can be a terminal effect of inhibition of oxidative phosphorylation? Anybody? If oxidative phosphorylation stops, then what happens? This is not compatible with life, because whatever we call respiration, okay, what we call respiration, oxygen taking inside, carbon dioxide going outside, this is breathing. What respiration is? This is cellular respiration, where oxygen is reduced through electron transport chain, or electrons, they are transported to the oxygen for the formation of water. So oxygen is used for the formation of water, and during the oxidation of substrate, carbon dioxide is evolved, as you know, in case of TCA cycle, that is liberated out. So oxygen is not converted to carbon dioxide. Whatever carbon dioxide previously been trapped by the plants in the form of carbohydrates, that is liberated, and the energy is liberated in the form of electron. Finally, oxygen, it forms water. This is metabolic water. This is cellular respiration, and this cellular respiration is used to trap the energy. And if this stops, everything stops, because whether you voluntarily do some work or not, there is enormous quantity of ATP that we require per day. Do you know how much ATP we require per day? If you weigh 70 kilograms, then you require 35 kilograms of ATP per day. So half of your body weight is the requirement of ATP per day by a normal healthy individual. So this occurs due to the cycling. This is not like you are putting on 35 kilograms per day. This doesn't occur because once it is been used up, it is converted to ADP. ADP is again converted to ATP. So this cycle goes on. You don't put on weight, but it is estimated that around 35 kilograms of ATP is required. That means half of the body weight is the turnover of ATP per day. So that is enormous quantity of ATP that we use per day. And you can calculate how much calories are required for generation of that particular ATP, and that is only 30% that has been used. So you have to make it 100% that is required by a normal healthy individual for the formation of ATP. Then there are rest of other things you have to do. So whether you do voluntarily work or not, you require ATP. Some of the organs, okay, they are continuously working and they require ATP, whether you use it or not. Like brain, for example. Intelligence is different thing and energy is different thing. Intelligence in biochemistry, intelligence in anatomy or physiology is different thing. But all of us, whether he is a medico, whether he is not a medical student, he uses same energy, or his brain uses same energy, same quantity of energy. I am talking about a normal healthy individual who normally thinks. Okay. So maximum amount of energy that is given up by the ATP is used by the brain, kidney, GIT. So these are the organs. Then muscle, liver, they also require enormous quantity of energy. So kidney, suprarenals, brain, these are the, or heart, these are the organs which require enormous quantity of energy. And you know that the number of mitochondria are more in case of heart, and if there is block in the respiratory chain due to the lack of oxygen, the particular tissue dies. That is called, in the case of heart, it is called as myocardial infarction, because it requires continuous supply of energy. So that is the quantity of energy, or that is the quantity of ATP that we require daily for our day-to-day activities. This is the ultimate thing where energy is captured. Now, apart from uncouplers, physiological or chemical uncouplers, there are certain poisons or chemicals which inhibits the electron transport chain. The name F0 itself indicate that these units, they are sensitive to oligomycin. So oligomycin, it inhibits at complex F0. It inhibits oxidative phosphorylation. So complex wise, complex 1 is inhibited by fish poison called as protenone or pyrazidine. Complex 2 is inhibited by malonate. Complex 3 is inhibited by antimycin. There are or BAL, British anti-leviside. Complex 4, that is cytochrome oxidase, is inhibited by carbon monoxide, H2S, and cyanide. and this is irreversible inhibition. Okay. So this is the final step. If inhibition occurs at first substrate level, that is at complex 1, there can be a utilization of complex 2 onwards. See, if the first complex, that is NAD dehydrogenase or NAD dependent dehydrogenases, they stop working, or whatever NADH is liberated, that is not utilized by the electron transport chain, but succinate dehydrogenase or from fatty acid oxidation, FADH can enter at the level of complex 2 and goes on, oxidation goes on. But this is not you can say unlimited thing. These are limited things. Finally, any inhibition to the electron transport chain will definitely hamper the oxidative phosphorylation. But for that matter, the last one, the inhibition of cytochrome oxidase by cyanide, is very grateful. So immediate death occurred due to the cyanide poisoning. Okay. Because cyanide, it inhibits cytochrome oxidase. The last component, and if last component stops working, then electron cannot flow. Okay. Because there is cycling, recycling and cycling again and again. You require an acceptor when somebody donates. So देणाऱ्याने देत जावे, घेणाऱ्याने घेत जावे. घेता घेता एक दिवस देणाऱ्याचे हात घ्यावे. म्हणजे काय? Whose poem is this? अरे वाचत चला. देणाऱ्याने देत जावे, घेणाऱ्याने घेत जावे. घेता घेता एक दिवस देणाऱ्यांचे हात घ्यावे. मीन्स यू शुड बी लाईक दॅट वन, दाता. प्रत्येकाने दाता व्हायला पाहिजे, याचक नाही व्हायला पाहिजे. Somebody who asks, but whatever, if somebody wants to donate, then somebody has to accept. If there is no acceptor, you cannot donate. Same thing occurs if cytochrome oxidase is, sorry, this is not this complex 4. If this is inhibited, okay, there is no flow of electrons. There is no flow of electrons from complex 1 onwards, because there is no acceptor. It stops over here. When this accepts from cytochrome C, then it gets oxidized. Otherwise, it remains reduced. If it is remain reduced, it cannot accept electron from this. If it is reduced, it cannot accept. So likewise. So everything stops, and there occurs immediate death in the cyanide poison. So these are certain inhibitors and uncouplers of electron transport chain. Now, the important thing is some of the reducing equivalent, they are synthesized in the matrix. Like in glycolysis, there is synthesis of NAD, or some of FADH2, they are synthesized, NADH is synthesized in the cytosol. And you know that inner mitochondrial membrane is not permeable to this. Then what happens? Then there are shuttles. There are two types of shuttles. One is called as glycerophosphate shuttle, and another one is malate shuttle. Glycerophosphate shuttle, it takes up NADH from the cytosol and convert it into the FADH2 in the mitochondria, because the dehydrogenase, glycerophosphate dehydrogenase in the cytosol is NAD linked, and in mitochondria it is FAD linked. So finally, how much ATP is synthesized, it depends. The NADH is synthesized in the cytosol, but if glycerophosphate shuttle is used, FADH2 is formed in the matrix or inside the mitochondria. So instead of 2.5, you get 1.5 ATPs. And then there is another shuttle called as malate shuttle, which is alpha ketoglutarate and malate, where malate dehydrogenase in the cytosol and in mitochondria they are NAD linked. So whatever NADH is formed in the cytosol is going inside the mitochondria, not as NADH but as intermediates like malate, alpha ketoglutarate inside, and again get converted back with the help of NAD. So NADH is converted to NAD in the cytosol. In mitochondria, this NAD is converted to NADH. So ultimately NADH is shuttled inside. So if this shuttle, this is called as malate shuttle. If malate shuttle is used, whatever NADH that is synthesized in the cytosol, it is directly converted to NADH in the mitochondria. So it depends which shuttle is used. If malate shuttle is used, there is no difference of calculation, or there is no difference. 2.5 ATPs will be synthesized inside, but if glycerophosphate shuttle is used, then 1.5. But mind well, for energetics, whenever NADH comes, we say that it is 2.5 ATPs, and whenever FADH2 comes, we say 1.5 ATPs, they are synthesized in the mitochondria. Okay. So this is all about oxidative phosphorylation. I just have to mention that there are certain disorders related to the oxidative phosphorylation or mitochondrial genome. They are chronic progressive external ophthalmoplegia. These are the names. Kearns-Sayre syndrome. What these are? Name indicates this is ophthalmoplegia. There occurs ptosis, okay, dual vision, weakness of muscles. It occurs over here. Kearns-Sayre syndrome. There is short stature. Okay. In case of Kearns-Sayre syndrome. Then there are conduction defects. So mitochondrial DNA is at fault, or sometimes the nuclear DNA is at fault. Then third is Pearson syndrome. Pearson syndrome, or it is also called as bone pancreas syndrome, where there occurs sideroblastic anemia as well as patient suffers from exocrine pancreatic deficiency. Later on, the features, if this patient survives in childhood, later on it seems or it has got the clinical features of Kearns-Sayre syndrome. Then there is MELAS. The name itself indicates it is MELAS. We can say that MELAS. It is myoclonus, epilepsy, lactic acidosis, and stroke like syndrome. MELAS, myoclonus, lactic, epilepsy, lactic acidosis, and stroke like syndrome. MELAS. Then there is MERRF, that is called as myoclonus epilepsy red fiber syndrome. So there are various names or various myoclonic epilepsy and red fiber. Red fiber is the muscle. It is seen under microscope, which are deficient in cytochrome oxidase. Okay. And mitochondria, they are functioning largely with the help of complex 2 activity is increased, complex 4 activity is not seen. So mitochondria, they are seen at the periphery of the muscle. All the muscle fiber or sporadically some part of segments of muscle they get attached. There is defect in that particular portion and there occurs proximal weakness. Okay, and muscles, and they are affected in these individuals. Weakness, fatigue is seen over here. Then there are other like leak syndrome is there, or you can say maternally inherited leak syndrome, where milestones are delayed, conductive deafnesses, dysfunctions are there. Some of the individuals, they suffer from deafness. Okay. And then there is popularly known disorder called as Leber's hereditary optic neuropathy is another disorder. Now all these disorders and much more disorders are there, like retinitis pigmentosa. This is one of the feature of mitochondrial mutations. So there is a vast different repertoire of picture of mitochondrial DNA or mitochondrial DNA diseases or OXPHOS. These are collectively called as OXPHOS diseases. So patient may suffer from any disease, but prominently if central nervous system is involved, then the patient suffers from nervous system dysfunction, or cardiac, if it is cardiac muscle, cardiac dysfunction is seen. Rarely the liver or kidneys, they are involved in the mitochondrial diseases. Now what is these are the some of the clinical features, like seizures are there, ataxia is there, dystonia is there, muscular weakness, fatigue is seen, ptosis is seen, all these persons, and popularly there occurs dimness of vision in second or third decade, and central vision is lost of one eye, followed by acutely or subacutely loss of vision in second. So person becomes blind in second or third decade of his life, and this is seen in running through families. And you know that most of the times if it is mitochondrial DNA, it runs through the mitochondria, maternal mitochondria. So previously it was very difficult to make out these patients. We just first diagnose and then come to know what type of mutation is there. There was no population genetics. Now efforts are there that population has to be screened for the mitochondrial oxidative phosphorylation diseases. So the newer type of treatment, like first is just palliative treatment to the genetic therapy or gene therapy comes up for the treatment of this OXPHOS diseases. I hope this is sufficient for you. Is there any difficulty? Okay. Thank you.